Good morning. I am Mushtaba Mansirpur, Research Manager at Plant Sciences. I'll be presenting work I did as a postdoc at the Cal Poly Strawberry Center on evaluating host resistant to Macrophomina crown rot in strawberry. Macrophomina crown rot or charcoal rot is one of the major soil-borne diseases of strawberry. So Macrophomina phaseolina is causal agent of this disease, which loves heat and high temperature. So we conducted this experiment at Cal Poly Strawberry Center, and I'm gonna talk about trial design at first, and then I'll talk about the method of inoculation that we used, and then I'll talk about evaluation method. At the end, I'll discuss the results. So we conducted this experiment in random complete block design or RCBD with 5 reps and 20 plants per rep. 4 reps were inoculated and 1 rep was non-inoculated or untreated check. This 5th rep or untreated check was bed fumigated with Ali 33 at 534 pounds per acre on October 7, 2019. Then we transplanted field on October 23, 2019 with 65 genotypes from 6 breeding programs including University of California, University of Florida, Driscoll's, Plant Sciences, Lassen Canyon, and California Berry Cultivars. Here you can see the location of Macrophomina field on Cal Poly campus, which is close to the Strawberry Center. And in this slide you can see the closer look of the field, and in the left side you see the inoculated side of our experiment, and at the right side you see the non-inoculated side of our field or fumigated side, which separated by 2 buffer rows. We inoculated the field 2 weeks after transplanting, and we used 5 grams of cornmeal sand Macrophomina inoculum. We put this inoculum around the crown and then covered that area with soil in order to increase disease pressure and induce the disease. We started to do drought stress from June 13th. And drought stress means withholding water, no water from Friday till Monday during the weekend from June 13th till end of experiment, which was on August 3rd. We evaluated field every 2 weeks from December, and I count number of dead plants, and those plants considered dead, which was 100% all foliage necrotic. So, and we also sample plots and beds by random to make sure they are dead because of Macrophomina phaseolina. In this slide you can see the way that we go through plot to plot, bed to bed, to read the number of dead and number of live plants. Here you see example of plots. Look, in this one at least 50% of plot dead, and in this slide you see some plants with brown leaf, but those are still considered live plant because they still have green foliage. And here you can see all these plants are dead. And here you can see the difference between diseased and healthy plant from crown section. You see the diseased plant crown section is discolored by Macrophomina, but the right one, the healthy one, is good. So here in this graph you see our results. On the x-axis you see plant mortality percentage, and on the y-axis you see the name of all 65 genotypes. So, and you see we have resistance at the bottom all the way to susceptibility at the top. So, and each one has an error bar which shows variation within rep for that specific cultivar. And if the bar is longer, means higher variation, and bar is shorter, lower variation, more consistency. So here at the bottom, you see Mariposa, UC Davis, CBC, and PS 9271 from Plant Sciences. So you see these four show 5% and low, and at the top you see Radiance, Ruby June, University of California, and Lassen Canyon, which shows 90 and above percentage mortality during this experiment. In this slide, you see the same genotype, the same experiment, but they sorted based on breeding program. And you see in each breeding program, they have both resistant and susceptible. So they have all of them in their material. At the end, we conclude that a wide range of susceptibility we have among genotypes and of breeding program, and we also have high level of resistance to Macrophomina crown rot, which is a useful source for breeders to transfer those resistant to new cultivars or their new popular commercial cultivars. At the end, all thanks California Strawberry Commission and NIFA SCRI grant to fund this project, and also all breeding programs involved in this project, including University of Florida, Driscoll's, Plant Sciences, UC Davis, Lassen Canyon, and California Berry Cultivar. At the end, thank you for your patience, and I'm ready to answer your questions.